Welcome back, folks. This is the chapter on renal physiology. The chapter is divided into three parts, the first of which looks at renal physiology. The second part looks at regulation of iron and water balance and how that's achieved. And the final section addresses pH balance in the body. Section A delves into the structure of the kidneys and their functions, as well as the involvement of the bladder. Most students remember a few functions of the kidney, but not the full complement. Beginning with the obvious, water regulation. The kidneys are known to regulate the water content of the body, as well as the inorganic iron balance. And that would include sodium, potassium, and a few other inorganic ions, as well as the acid-base balance, the pH, which also involves the lungs to a large degree. The kidneys remove two types of waste. Waste that's generated through metabolic processes within the body, and foreign substances entering the blood either through the digestive system or through intravenous injection, and sometimes even through the lungs. The kidneys are also equipped with the ability to generate glucose and release that into the circulatory system, thus helping regulate the homeostasis of glucose levels. Certain cells in the kidney can also produce and release hormones and or enzymes. And there's three types that we need to be aware of. The first is erythropoietin, which is a hormone that controls the production of red blood cells in the red marrow of the bones. The second is an enzyme, renin, and that enzyme controls the formation of angiotensin, which is a hormone that regulates blood pressure and sodium balance. Finally, the kidney is involved in the conversion of 25-hydroxy-vitamin D to 1,25-dihydroxy-vitamin D, the effect of which is to influence the calcium pool within the body, the calcium balance. The two kidneys are located outside the abdominal cavity, just below your rib cage. One is slightly lower than the other. In most humans, the left kidney is slightly higher in the abdominal space than is the right kidney. The kidneys have a blood supply that comes straight from the descending aorta and veins that carry blood back into the inferior vena cava, the main veins that return blood to the heart. Urine is collected in the pelvis of the kidney, and then it travels through peristalsis, through the ureter into the bladder, where it's stored until it's expelled through voluntary processes through another tube that's called the urethra. One of the most important and best ways to realize the functionality of a kidney is through its anatomy, its structure. So knowing the structural components of the kidney is key to learning its functions. So we should start with a figure such as this, 14.2. If you can remember all of these labels, that will be fantastic. So the cortex, which in my mind is the circumference, the cortex is the layer on the outer periphery of the kidney. And the medulla, the middle, will be the middle component of the kidney tissue. And then we have chambers, the papillae, and a very thin layer of connective tissue on the outside called the capsule, as well as the calyx, which leads to the renal pelvis. For the size of the organ, the kidneys have a large supply of blood entering and leaving, and that will become obvious shortly why that happens. The main anatomical and physiological units of the kidney are the nephrons. These are microscopic structures which we have to pay considerable attention to in order to learn the physiology of the kidney. An important anatomical observation is the close association between blood vessels and the nephron. These two types of tubes have a very important influence on each other, which along with hormones will dictate the type of urine the individual produces in the bladder. The urine can range from a very dilute watery mixture to a very concentrated and thick urine. By some estimates, we have about 2.5 million nephrons between our two kidneys, which translates to just over 1 million per kidney. It's also possible for an adult human to survive with just one kidney. We are going to devote a considerable amount of time now to looking at the microscopic organization of the nephrons and the blood supply intermingled with them. We begin our journey at the beginning of the nephron, at a small microscopic swelling known as a renal corpuscle. Beyond the corpuscle, there's a thin tube called the tubule that extends and meanders in several very defined orientations. A deeper investigation of the renal corpuscle reveals two intimately tied components. The first is a bed of capillaries known as the glomerulus. That is then tightly associated with the Bowman's capsule. The Bowman's capsule is the top part of the nephron. Figure 14.3 is very important. Not only does it give you the anatomical structure of two types of nephron in the kidney, it also relays the order in which fluids pass through different regions of that system. Understanding why evolution has come up with this exact arrangement of blood vessels and nephrons and collecting ducts is key to really understanding how the kidney has evolved to function in a beautiful way that it has. Investment of time on this figure is going to be rewarding later on. Where do we begin? First, it's important to realize that the two classes of nephrons are positioned in slightly different anatomical regions of the kidney's layers. The majority of nephrons are cortical nephrons. They have a small loop, but their general structure is very similar to the second class. The second class of nephrons, which are fewer in number, but have very long loops and very intimate blood supplies, are called medullary nephrons. We are going to spend the majority of our time understanding the medullary nephron because it gives rise to some unique features. The general organization of these nephrons is like the letter N. So they begin in one location, which is the corpuscle, then meander within the cortex, and thus called the proximal convoluted tubule. Then that takes a dive down, and that's known as the descending limb of the loop of Henle. Then we have a bend here, which is the loop of Henle. Then we have the thin segment of the ascending loop of Henle. Then we have the thick segment of the ascending loop of Henle. Then it forms a bend here, and then it convolutes a bit further. And this is the distal convoluted tubule. And then that joins the collecting duct, which it shares with numerous other nephrons that are attached in various branches. Basically, what we just did is we followed this pathway. This indicates the pathway may be linear, but there are two bends. There's a bend down here, and there's another bend here before it goes down south. With respect to the blood supply, there's two important regions of note. The first thing is blood enters the kidney through these arteries, and then they branch, and the first thing they do is feed the renal corpuscle. So this is called the afferent arteriole. Afferent means coming in, and as soon as it gets into the Bowman's capsule, that artery starts dividing into capillaries, and that is the glomerulus. As the capillaries coalesce again, they form another arteriole called the efferent arteriole. The efferent arteriole doesn't go out into the vein, it dives down and it follows the loop of Henle into the deep medulla. Then the blood supply loops around and comes back up, following again the loop of Henle. And it's then that it joins the venules and it begins to leave the kidney. So we have two capillary beds separated by arterioles. The corpuscle, just like other parts of the tubule, is a fascinating region with its own peculiar anatomy and physiology. The first thing is the lining, the cells lining the capillaries, the endothelial cells, they don't form a complete and solid wall. Instead, they have large gaps which allow solute-rich fluid to pass between them. That condition, which is used in a couple of other places in the body, is called a fenestrated capillary. The purpose of this design is to encourage the flow of fluid out of the blood, the plasma, without allowing the cells, the red blood cells and the white blood cells, and proteins of a significant size. So basically, most of the large proteins and the blood cells are prevented from passing from the blood, which is under pressure, across into the Bowman's capsule. To further encourage the filtration mechanism, the other side, the Bowman's capsule itself, has its own lining of cells, and those cells have special properties too, in that their cells have special shapes called podocytes that also generate large gaps to encourage filtration. These are scientifically known as filtration slits. In addition, the arterioles feeding the capillaries of the glomerulus are lined with modified smooth muscle cells called mesangial cells, and these, as we'll see shortly, regulate blood pressure and thus can influence the production of filtrate. The top half of figure 14.4 demonstrates this. So in pink we have the glomerular capillaries. In red we have the two arterioles, the efferent arterioles which bring blood in, and the efferent arterioles which take blood away. Then we have the yellow structure. These are the cells of the Bowman's capsule. The bottom half of the figure shows both a magnified view demonstrating the pink podocytes with their filtration slits and the red endothelial cells with their fenestrations. The very bottom shows a cartoon of the two-dimensional relationship between the endothelial cells of the blood vessel at the bottom, the basement membrane, and the cellular podocytes that are part of the Bowman's capsule. The design of the two sets of capillaries is very important. So as blood comes in, we already mentioned that it first flows into the glomerular capillaries, then it travels into another set of capillaries, and these are called the peritubular. If we go back to the figure here, we can see these are the glomerular capillaries and these are the peritubular. These are also known as the vasa recta. The vasa recta. The purpose of the glomerular capillaries is to permit filtration. The amount of filtrate produced can be directly influenced by both the local blood pressure and the overall blood pressure of the circulatory system. With respect to the local blood pressure, each arteriole that enters and leaves the glomerulus has smooth muscle, and that smooth muscle can be altered, contracted to reduce the diameter of the capillary, therefore reducing flow. This very localized blood pressure is known as the glomerular blood pressure, PGC. If this blood pressure is high, a lot of filtrate is produced per unit time, and the converse, if this blood pressure is low, less filtrate is produced per unit time. In a typical day for an adult male, between the two kidneys, about 180 liters of filtrate are produced, an amazing amount. But most of this is reabsorbed soon after the corpuscle, whilst it is flowing through the proximal convoluted tubule. And this fluid ends up in the peritubular capillaries that follow the loop of Henle. This slide summarizes what we mentioned earlier about the two classes of nephrons. Another important observation is that there's a tiny region of cells in each efferent arteriole that are special, and these cells are known as the juxtaglomerular cells, which are mechanically gated. So when they experience a reduction in stretch, they release an enzyme called renin, and that enzyme has an indirect influence on sodium balance and blood pressure in the entire body. But wait, there's more. There's another set of cells which don't belong to the afferent arteriole. They actually belong to part of the nephron. So as the ascending loop of Henle makes a turn and becomes the distal convoluted tubule, in that region there's a few cells known as the macula densa. These cells can sense the fluid passing in the tubule, and they can then make adjustments to regulate the sodium balance and the blood pressure as well. So the two sets of cells are next to each other, and they're talking to each other, and they're regulating sodium balance and blood pressure. And the combination of this region has a name. It's called the juxtaglomerular apparatus, the JGA. Figure 14.5 highlights the two classes of cells. The juxtaglomerular cells, the JG cells, are in green, and in purple we can see the macula densa. You can see the close approximation to each other, and that's important in one influencing the other. We'll readdress this in more detail shortly. This and the next couple of slides reveal some important forces at work when it comes to the kidney. The three that we have to learn immediately is the force driving the production of the filtrate. That's called the glomerular filtration force. Then an alternative way to remove material from blood vessels is to secrete it across these cells lining the tubule, and that's called tubular secretion. And then finally, you can reabsorb material back out of the tubule into the blood system, and that's called tubular reabsorption. So these three forces may be working, may not be working, or may be working in some combination on any single component that the kidney is trying to regulate. The general equation is that the amount of a substance excreted by the kidneys in the urine is equal to the amount that's filtered plus the amount that's secreted minus the amount that's pulled back through reabsorption into the blood. For some substances, the pathway is indicated on the left-hand panel. So here we have something that is not filtered much, a tiny amount is, but it's mostly secreted later on by the interaction of the vasorector with the nephron. The center hypothetical shows a purple substance which is filtered to a small degree, but is then mostly reabsorbed, so that a tiny amount is secreted by the kidney. The last panel shows a substance which is filtered to a small degree and is entirely reabsorbed back into the blood. And the last one will be an example of glucose. Under normal circumstances, that is in non-diabetics, the amount of glucose that's filtered is easily reabsorbed, so that no glucose appears in the excreted portion of the urine. However, for a diabetic, the process of filtration is enhanced so that they have a lot of filtrate, but it can't all be reabsorbed because there isn't enough time, and that leads to glucose being detected in the excreted urine. Amazing. The textbook likes to use the word bulk flow. Bulk flow is the movement of fluid under pressure, hydrostatic pressure, across a filtration barrier. This is not the same thing as osmosis. Osmosis is just the movement of water across a membrane. But here we're looking at the water plus some low molecular weight substances that are dissolved in the water, and that can include some small polypeptides. Thus, by using bulk flow, because of blood pressure in the afferent arterioles and the glomerular capillaries, plasma is forced across the filtration slits into the Bowman's capsule. And because of the number of nephrons, 2.5 million, the amount of filtrate produced can be enormous. As we alluded to earlier, a typical result for this filtration rate is about 180 liters per day versus the normal capillary beds in tissue, which only filter about 4 liters per day. Interestingly, the opposite force is the osmotic force generated by the presence of the proteins that are left behind in the blood plasma, and they are the main way in which you get the fluid being pulled back out of the nephron, the tubule, into the blood vessels. So that's part of the reabsorptive force that we talk about in various places in the human body. Thus, the degree to which filtration takes place in the glomerulus, the glomerular filtration, is based on several forces and their mathematical arithmetic. If you look at the arrows, the thickness of the arrow is an indicator of the size of the force. In this particular example, the center arrow tells us that there's a large force, the blood pressure in this glomerular capillary, that is forcing the fluid part of the blood, the plasma, to be filtered. But there's also two back pressures. The first back pressure is easy to understand, and that's the osmotic force due to the extra proteins that can't leave. So the proteins that are left behind in the capillary, they have a pulling force back on that fluid. And there's a second force here at the very top, the PBS. And that's the pressure of the fluid in the Bowman's capsule building up. And as that fluid builds up and it can't leave fast enough down this pathway here, it has a back pressure, and that's the back pressure there. So if you take all the values and you do some simple arithmetic, and in this case it's going to be the pressure, the blood pressure, minus the back pressure, the osmotic force, minus the back pressure due to the pressure in the Bowman's capsule. In this case, the 16 millimeters of mercury is the net force driving the production of filtrate. One can see that by increasing the blood pressure, the degree to which filtrate will be made will increase. And on the contrary, by reducing blood pressure in this section, the amount of filtrate that's made will be reduced. We do know that the arterioles that are feeding the glomerulus capillaries, they do have smooth muscle wrapping around them. So by constricting the smooth muscle on one side versus the other, we can then control blood pressure from both sides. So let's just look at this column here. So in this column, we are having blood flow from the left to the right. And if we constrict the afferent arteriole, the one that's coming in, then naturally blood pressure will fall on this side of the constriction. Therefore, the rate of filtration will be severely decreased. Now, if we do the same thing, but this time we dilate the efferent arteriole by relaxing its muscles, we will also have a reduction in blood pressure. And that will also lead to a reduction in filtration. On the contrary, if we constrict the efferent arteriole, we will then cause a backlog here that will lead to a greater filtration rate. Or we could do something different, is dilate the afferent arteriole, the afferent AA, and that will have a similar effect by allowing more volume of blood to reach this point and also increase the rate of filtration. So by adjusting things in different parameters, the nephron will receive different amounts of filtrate. Although we can't list everything that the kidney does, because there's so many substances that it can secrete, we have some common ones here. So let's go back to that 180 liters per day of water that's made by the kidneys. Well, luckily for us, 99% of that 180 is reabsorbed. Only about 1.8% is available to be excreted. And that can vary depending on your level of hydration. How about sodium? Well, this is in grams now, not liters. So 630 grams of sodium are filtered every day. And even more of that is reabsorbed, 99.5%. So only about 3.2 grams of sodium is lost on a typical day. And again, that's dependent on your diet. 180 grams of glucose are filtered each day, and 100% of that is reabsorbed. Therefore, 0% of glucose should appear in the secretions. And urea, 54 grams of that is filtered. Most of that is secreted. Only 44% is reabsorbed. The rest, which is waste, is eliminated. For material to move from one location, the lumen of the tubule, to the blood vessels, it cannot pass directly between them because, as we know, blood vessels are lined with cells called endothelial cells, and the tubule itself, the tubular lumen, is surrounded by endothelial cells too. So these cells in brown belong to the tubule, and they have tight junctions right there to prevent small molecules from passing between them. Therefore, everything has to be directed through the cell cytosol. And we have on the surface here certain receptors, ion channels, pumps, and we may have a different combination of those same elements or different elements in terms of receptors and pumps and ion channels here. So these cells can discriminate in what they pick up and what they then pass to the other side in both directions. And that's going to be very important as we learn about the functioning of the kidney. Sodium is an important commodity for land-based creatures. So plants and animals, we value sodium because wherever sodium goes, water follows. That's the general remit, right? If you control the sodium, you will also control the water that follows the sodium. So understanding that is very important. The nephron is designed to reabsorb sodium all along its length, except in one place, and that is the descending loop of Henle. That's a very important concept, and we'll come back to this again and again in the next few minutes. But first, we need to revisit some cell biology concepts. The movement of sodium is highly influenced by something known as the sodium-potassium pump, otherwise known as the sodium-potassium ATPase. This membrane protein is very important. It's present in every cell in the body, and its purpose is to move sodium and potassium against their concentration gradients. The ATPases are located on the basolateral membrane. That's this membrane on this right-hand side of these tight junctions. This continuous activity generates a low concentration of sodium inside these tubular epithelial cells. This allows the apical surface, the surface facing the tubular lumen, to then suck in sodium from the fluid, the filtrate that's passing through. By regulating the behavior of these sodium ion channels on the apical side, the system can decide how much sodium should be withdrawn through recovery, reabsorption from the filtrate, therefore changing the composition of sodium. And through coupling, either antiport or symport, other ions like potassium can be either eliminated or recovered, therefore changing their concentration in the filtrate. And as we see later, the concentration of protons, hydrogen ions, i.e. pH, can also be regulated through a similar mechanism. So it should come as no surprise that glucose and most amino acids and many organic substances are transported by being coupled to the movement of sodium. The graph in figure 1411 shows the relationship between the glucose concentration in the plasma, measured in milligrams per deciliter, versus 3 different states in which glucose can be moved. So one of these is the filtered rate. The second is the excreted rate in the green line, and the last in the blue is the reabsorbed rate. A superficial analysis of this data can be made by comparing a normal person with a diabetic. The normal blood glucose levels in a healthy adult are just about 100 milligrams per deciliter. So at that level, all the glucose in the blood plasma is secreted, but in the proximal convoluted tubule, it's all reabsorbed because there's enough time and enough cells in contact with the filtrate to reabsorb all that glucose back into the blood. So in a healthy person, we should not see any glucose in the blood. However, in somebody who's severely diabetic, say 400 milligrams per deciliter, their level of glucose would be so great that the system would have no capacity to absorb all that in the time that it's given as the filtrate is moving through the tubule. Therefore, the level that's secreted will begin to increase, and that's indicated by the green line. The conclusion is that our tubules have a limited capacity to reabsorb glucose back from the filtrate. Anything beyond that will then be eliminated. The importance of tubular secretion cannot be overstated. For some chemicals, such as hydrogen ions, potassium, and organic anions, their movement from the blood to the urine is by tubular secretion. In general, tubular secretion is an important mechanism for getting rid of drugs and the metabolites of drugs, for eliminating undesired substances or end products of metabolism that have been reabsorbed by other processes, and for the removing excess potassium, and of course, and controlling the pH of the blood. We stated earlier that kidneys can make glucose. This is actually done by the tubule, the renal tubule cells that can synthesize glucose when other systems in the body are running low on glucose. And they can release that glucose into the blood to keep the brain happy. The way they do that is they take other organic substances like peptides and proteins, and they absorb them into the cells, and then using their internal biochemistry, they convert through a sophisticated part of the cellular respiration into glucose, which is then released into the blood. We keep using the word regulation. You can substitute the word control in its place. So control of membrane channels and transporters. So this is the key to how the kidney works in the second half of the tubule. So the first half of the tubule is more or less working by default based on the rate of the filtrate. But the second half of the loop of Henle can be more regulated by hormones and other factors in the general circulation. So reabsorption and secretion takes place through transporters and membrane channel proteins, and they can be then turned on and off depending on the needs of the body. This last statement is important for students to realize. The cells lining the nephron are not all the same. They change their identity and their biochemistry as one migrates along the nephron. So the majority of reabsorption takes place in the proximal tubule, the proximal convoluted tubule, and the early part of the loop of Henle. Further along, in the proximal tubule and the loop of Henle, reabsorption can be extensive for certain solutes and for water. So here, water is recovered so that the amount of water that's left in the filtrate is relatively small. In the more distal parts of the tubule, that's when the fine-tuning takes place with the help of hormones and other types of substances. The last concept under this umbrella is that of renal clearance, which is defined as the volume of plasma from which that substance is completely removed by the kidneys per unit time. And here's the equation that relates to it. So the clearance of that substrate is equal to the urine concentration of that substrate to begin with, the urine volume per unit time divided by plasma concentration of that substance. The standard substance used by physiologists has been inulin, being a form of dietary carbohydrate. Its important property is that it is freely filtered by the kidney, but is not reabsorbed nor secreted. So it's an ideal standard molecule. Therefore, the profile for inulin will look like this, where the only activity is filtration. There is no secretion or reabsorption. Micturition is the process of urinating. The process involves the interplay of smooth muscles surrounding the bladder, as well as straightened muscles at the external urethral sphincter. The bladder is under three types of external regulation: the parasympathetic nervous system, the sympathetic nervous system, and also the motor neurons belonging to the somatic nervous system. Thus, micturition is both a reflex involving both voluntary and involuntary control. Table 1413 nicely compares and contrasts the different areas that are under regulation. Incontinence, the medical term for the involuntary release of urine, is due to many reasons. The most obvious is stress incontinence due to sneezing, coughing, or exercise, where pressure is placed on the bladder by the surrounding tissues. And urge incontinence, which is the desire to urinate regardless of external conditions. Women, because of their hormones and physiology, have a more difficult issue with this problem. We'll pause the video here and break it up into two parts.